Retina Rounds podcast number 8. As part of our Giants in Retina series, Retina Rounds is honored to welcome Dr. Barry Kuppermann. For over 3 decades, Dr. Kuppermann has been an innovator and pioneer in the field of ophthalmic drug delivery, having made transformational contributions in the management of both infectious and inflammatory ocular disorders. In addition, Dr. Kuppermann has been an academic leader. As chair of the Department of Ophthalmology at the University of California, Irvine, and director of the Gavin Herbert Eye Institute, Dr. Kuppermann has built one of the premier retina divisions and ophthalmology departments in the country. In this episode, Dr. Kuppermann talks about the guiding principles in his professional life, including intellectual curiosity and integrity, the role mentors and collaborators have played in his career arc, and the values that have kept him excited and engaged in academic medicine. Let's check it out.
Okay, well, welcome to the weekly Retina Rounds podcast and our Giants in Retina series. This week, we're privileged to have with us Dr. Barry Kuppermann, Professor Barry Kuppermann, Chairman Barry Kuppermann at the University of California, Irvine Eye Center. I forget the exact title of the Eye Center, Barry. What is it?
The Gavin Herbert Eye Institute.
That's right. And tell our listeners who Gavin Herbert was.
Gavin Herbert is the founder of Allergan. As a graduate of USC School of Pharmacy, he and his dad set up a compounding pharmacy above a burlesque theater in downtown LA and came up with an eye drop. And from there, it flourished into Allergan, which moved to Orange County. And amongst the most things Allergan was most famous for, ultimately was commercializing Botox. So for all you Botox users out there, it's all originally from Allergan. Allergan eventually got sold. Now it belongs to AbbVie. And Gavin Herbert, the founder, age 93, is still a brilliant person, and I have lunch with him every other week or so.
Yeah, and aren't you lucky to be around Gavin, who's one of the greats in our field and a true entrepreneur and has given so much to society, not the least of which is endowing the Eye Center. Well, thank you again for joining us today on Retina Rounds. It's such an exciting time to be a retina specialist and to be learning about retina. Barry, I thought I'd start this chat off by asking you what you feel has been the most 2 or 3 transformative innovations that you've been a part of or been witness to during the arc of your career.
Well, thanks, Steve. And again, it's a pleasure to be here. And the arc of our career, since we're contemporaries, has been quite long now. As you know, the early phase, so I've had 2 sort of, in my mind, phases of my career in a certain sense, all of which I've spent here at UC Irvine, much like you had a long career at UCLA. And again, we were very paired up. You were the chief of the retina service at UCLA. And for most of my career, I was the chief of the retina service here at UC Irvine, then became department chair 8 years ago with the sad passing of Roger Steinert, who I still adore, passed away from glioblastoma. He was a wonderful chair, and I took over after him. But in the first phase of my career, I was sort of the serendipity of life. As you know, I had a funny background. I went to Berkeley as an undergrad, went to Caltech and got a PhD, just interested in science, and in this case, learning at the synaptic level. So I studied neuroscience, and then I studied the visual cortex as the model system. And from there, I thought it'd be interesting to go into medicine and become an ophthalmologist. But when I arrived here at UC Irvine, actually before that, when I was a resident at USC, again, Doheny USC, another change in life as Doheny is now UCLA, part of the UCLA empire, I took on a passion for the ocular complications of AIDS. My first rotation at Doheny as a brand new resident was with Narsing Rao, who was both a pathologist and the uveitis specialist, and he was responsible for running at LA County the AIDS clinics. And this was the 1987, where AIDS was a scourge, and it was a dilemma. It was everybody that was our age, with all the similar interests, men of our age, similar interests, but they were gay and we weren't, and they were dying and going blind and we weren't. And it was, to me, an epiphany. I discovered belatedly why I had become a doctor. It became my motivation, my reason for being. And so I was devoted to that throughout my residency and in my fellowship years. And part of why I came to UC Irvine was that there was not a specialist in the ocular complications of AIDS. And I devoted a lot of my early phase of my career to clinical development of treatments. I became interested in drug delivery because of that. I was the lead investigator on the Gancycler implant, not something I developed, but something I helped commercialize was the lead investigator for that. I was fresh out of fellowship, and I became my first clinical trial ever. I was the national leader of that clinical trial, and that, which is still kind of amazing. And presented the data when I was 3 or 4 or 5 years out of fellowship. That was me and Dan Martin, also fresh, relatively fresh out of fellowship, presenting the data to the FDA. So that was a dramatic thing. That to me still was the most powerful and moving thing I've ever done professionally in my life. I used to, back in that sad era, I used to make rounds, not only to every hospital in town, but to my patients' homes. I would have a little bag and I'd show up with ganciclovir injections that I would administer to my patients in their homes on a regular basis because they were too sick to come into the office sometimes, or to whatever hospital they were interned at, because they had, again, end stage AIDS and couldn't make it to their appointment. So that was, to me, amazing. And then, as a lesson from the pharmaceutical industry, which I've happily partnered with, I understand the complexities and nuances, that it's not all, golden and good. You have to manage it properly, but I want to give credit where credit's due. They conquered the AIDS epidemic. It became, with advances of highly active antiretroviral therapy and the advancements in treatment for the ocular complication of AIDS, CMV retinitis in particular, now it disappeared as a problem. There was a period of time back in the 90s that I think Bill Freeman, another mentor, wrote an article where he predicted that, based on statistics, the number one cause of retinal detachments was going to be, never achieved that, CMV retinitis related retinal detachments. That's how common the disease was and how the complications were manifesting themselves. But again, through all the advances, through the collaboration with the pharmaceutical industry, we're able to conquer that disease, and AIDS is now a manageable chronic disease, and CMV retinitis almost never appears anymore, where it used to be in our clinics all day long, every day. So that was a huge and important thing. It led, when I came to UC Irvine, and again, why I came here was there was not a clear somebody that was interested in the ocular complications of AIDS, and that's when I became involved with the ganciclovir implant trial, the Vitrasert trial. I also set up a drug delivery unit here, and I developed got a joint appointment with the Department of Biomedical Engineering to develop drug delivery systems, for which we have patents in. So that was in the early decade or more of my career, clearly the thing that motivated me the most and was most passionate and most meaningful. And it was a remarkable era for all of us physicians.
Barry, that is an amazing story. And can you share with our listeners, our audience, the specifics of the ganciclovir implant? How did it work? And it was a depot with a membrane, but talk us through how it worked. Sure. And what the barriers were to getting this popularized and proven efficacy and safety and so on.
Okay, because I'll get to then the second phase, which is, of course, the one that we all share together, which is our 20th anniversary is coming up in a few weeks when we celebrate ASRS in Long Beach, halfway between our two homes. That'll be the 20th anniversary of anti-VEGF therapy administered on a regular basis. So I'll get to that as my second part. But back to the ganciclovir implant. Again, in that era, the treatment of cytomegalovirus retinitis, an opportunistic infection that happened only if your AIDS status, your HIV status was so advanced that your CD4 count, the population of lymphocytes in your body, was so low that basically you were in the last year of your life. This was a late manifestation of AIDS, to remind people about it that didn't participate in that era. And so as your immune function declined, you were subject to more and more opportunistic infections. And one of the last ones that anybody would get was cytomegalovirus retinitis, which is a reactivation of that virus that is in all of our bodies. 85% of the population has CMV in them, but it reactivates because they don't have any immune defenses against it. And that was leading people to go blind. And these were these 35-year-olds that were in our offices begging us not to let them die blind. That was their one plea. And so what happened is that there was intravenous therapy of ganciclovir that was being administered. It was a huge amount of drug, 5 milligram per kilogram twice a day until you got under induction, and then maintenance therapy of 5 milligram per kilogram once a day. And then we started giving intravitreal injections way back when, in that era, on a weekly basis of either ganciclovir or foscarnet, the other drug that was available. And then along came this small little company made up of ophthalmologist and a pharmacologist, controlled delivery systems. Paul Ashton, Tom Smith, and Andy Pearson were the founders of that. And they developed this Gancipher implant, a little pellet that contained a total of 5 milligrams of drug, surrounded by a permeable membrane, which was then surrounded by an impermeable membrane with a break around it in a ring structure with a tab that you would then trim the tab, and you would go into the operating room and make an incision infratemporally in the sclera in the vitreous base in that pars plana area, about a 5 millimeter incision, and you would trim the tab and punch a hole in it, have a suture set up, and you would suture it into the wall of the sclera, and it would deliver drug over 8 months. And that 8 months was the bulk of their remaining life. Most of these people sadly only had about a year or so of life, but I've had many of those that lived longer and they got multiple implants, but 5 milligrams of drug controlled their cytomegalovirus retinitis better than anything else. And the drug levels in the eye were fourfold what you could ever achieve with intravenous therapy. I've presented on this. Intravenous therapy over 8 months, assuming the 5 milligram per kilogram induction phase, followed by the 5 milligram twice a day, followed by the 5 milligram per kilogram daily maintenance phase, was about 100,000 milligrams of drug over 8 months, compared to 5 milligrams in the entire pellet, and that got a fourfold higher load inside the eye that showed the marvel of local therapy and drug delivery that we're now emerging onto, ironically, now 25 years later, in our modern era of anti-VEGF therapy. For 20 years, we've been injecting drugs into the eye, but now under development is drug delivery systems more and more. Ironically, one of them, and to harking back to controlled delivery systems and the originator of the Vitracert, they also developed the Reticert with fluocinolone and acetonide. Then they developed the Utique and Illuvian, which they then licensed out to others. That company changed name from Controlled Delivery System to Cyvita. Now they're called EyePoint, run by our friend Jay Duker, and they have now developing their own implant now, the DuraView, which is a TKI, which is biodegradable. So you see the arc of this career actually bridged 2 zones of drug delivery: AIDS-related ocular complication of AIDS drug delivery with the and then the modern era that now Ocular Therapeutics has a drug delivery system with their Axpaxli that has another TKI. That one's axitinib that's injected as a hydrogel into the eye that's being tested. And EyePoint is being, and that, of course, that company is run by other friends, Praveen Dougal, Jeff Heyer, Peter Kaiser, Nadia Wahib. And then again, the controlled delivery systems to Cyvita to EyePoint, run by Jay Duker et al., has a pellet that is a biodegradable system. They're both biodegradable with a different TKI vorolanib in the modern era of anti-VEGF therapy. So these 2 arcs of this career, ironically, are linked up through the drug delivery story.
Well, there's some fascinating points that you really glossed over, one of which is huge controversy at the time of whether or not an eye could sustain or withstand multiple injections into the pars plana, into the vitreous. And you and your group really showed that. And second of all, the importance of local delivery versus systemic delivery. I remember very clearly when there was a moment when the first anti-VEGF agent had come out, Macugen, and there was a big push for systemic Avastin delivery. And you and I were at a meeting in Miami where our friend Phil Rosenfeld was talking about IV Avastin, and you just about had a stroke because of your experience with the dominance of local therapy over systemic therapy, and that very quickly morphed into local Avastin, even though there was some question about whether or not Avastin was too big of a molecule to make it through the neurosensory retina and down to where the angiogenic activity was. So those were really pioneering times, and people might not realize just how remarkably prevalent CMV retinitis and detachment was. I mean, in my career, there was a time where we were doing 5 to 10 detachments a week CMV just at our center, which was one of multiple centers in our greater metropolitan Southern California area, that were all doing a similar volume of detachment. So it was an absolute tidal wave of pathology. And you're exactly right. And that's another point that you touched on, which is that people tend to care more about their vision than their life. We've all had the experience where a cancer patient will say, I want to see for the duration of my lifespan or health span, rather than I want to live longer blind. And that really came home to roost through the course of your work there. And so then you were going to touch on your experiences with drug delivery, and for our audience, Barry's very humble. His contributions to drug delivery are legion, but let's talk a little bit about that, about the next great sea change in our field, which was the pharmacologic control of hyperpermeability and angiogenesis with agents that block angiogenesis. And really, it is the 20th anniversary of commercialization. Really, Macugen, I think, was approved in '03, late '03, early '04, with David Geyer and Tony Adamus and our group at then iTech. But we were giving anti-VEGF agents in clinical trials all the way back into the late '90s, and so we saw patients responding to this. They were masked, of course, but we saw patients responding to this, and we knew, and even our coordinators knew. I'll never forget having a number of clinical trials open at the time, different variants of photodynamic therapy and others, and submacular surgery trials, and my coordinator coming to me and saying, we should do more of this. Mm-hmm. So tell us, Barry, your experience with the development of the anti-VEGF revolution.
Well, as you point out, it was at a very exciting time. Bridging these, again, the local therapy was the theme, and we learned that back in that CMV retinitis era, where literally we're giving injections weekly. That's what it took before we had the drug delivery system that lasted at least 8 months. And just to finish up with that, the week when you were doing all those retinal attachments, as I was too, I had days where my operating room, I had to have extra operating room days. I'd had days where I'd put in 15 Ganciclovir implants back to back. It was like cataract surgery. I just had a stream of patients coming in for surgery that day. I had days just for Ganciclovir implants. But back to then the modern era of anti-VEGF therapy and local therapy, as you point out, Macugen, which you played a big role in. I mean, you were very participatory with iTech, pegaftinib, and that was very exciting. With a caveat, we had photodynamic therapy, and then we had Macugen. These were incremental steps, but the science of Macugen was super impressive, but the net results were a little bit less than we wanted. It was good, but not great. We were very happy to have it. Then along came, that's where that seminal year that I'm referencing, July of 2005, at the ASRS meeting in Montreal. There on the podium was Phil Rosenfeld presenting his Avastin data. Next to him, next present was Joan Miller presenting the Marina data about Lucentis. And of course, it was also due reference was moderated by Carmen Pugliafito up on the podium. And that was also an interesting meeting, because there we were in ASRS, all together in a room, as we always are. I love ASRS because it's like the biggest meeting we go to that is always in one room only. But we looked around and said, who are all these people? Because it's the first time we saw a room full of retina specialists and investors. The financial community was in that room. It was a game changer, because suddenly we were learning about the glories of Avastin, the glories of Lucentis, even though we'd been in the trials, and those results. And that was also an important year for another reason, another technology. We all got our first OCT machines that year, the temporal domain Stratus OCT by Zeiss. That was before spectral domain. And so in that year, that summer, we got an amazing drug that we all started using right away, Avastin, that finally we were really now, it was a step up from PDT to Macugen, now to Avastin. And honestly, everything since Avastin has been a minimally modest incremental benefit. They're definitely better, in many ways, but not dramatically in the way that Avastin was from the others. Similarly, we suddenly got OCT that we never had before, and that was a huge change. So we had an amazing drug and a way to monitor the disease, and that was revolutionary. But we would have never believed in 2005 or in 2006 or in 2007, as Lucentis came along as well, that 20 years or 18 years later, we'd still be more or less doing the same. We thought that by now we would be far along to something else. And that's what you're working on and others are working on to get us to that something else, because we're still loving what we can do for our patients, but we want to do even more now. And we're still up against this kind of ceiling of drug delivery, the duration, and all those things. So it's been a remarkable era where we've stopped. Me honestly, when you and I trained, as you know, I hate to be one of those old guys, but we viewed ourselves primarily as surgeons, and we still think of ourselves as surgeons. Honestly, we've all become much more medical retina specialists than surgical retina specialists. Medical retina has dominated our field because of all the good we can do with these injections and treatments that started with pegaptidin, Macogen, and then moved on to bevacizumab, Avastin, ranibizumab, Lucentis, and then aflibercept Eylea, and then now on to, of course, faricimab and aflibercept high dose. So it's been a remarkable 20-year, 23-year stretch that we've transformed the treatment for our patients, not, of course, just with neovascular AMD, but with diabetic macular edema, diabetic retinopathy, retinal vein occlusion, myopic maculopathy, so many things we've been able to achieve through this local delivery of these amazing drugs.
Yeah, Barry, you're also the king of steroids, at least in my mind. And I'd love for you to take us through a little bit of the work that you've done on long-term steroid delivery.
Yeah.
It's really been an amazing story. You know, for those of you in the audience listening, steroids were met with a lot of concern because of the well-known side effects and potential for atrophy and immunosuppression and IOP elevation. And Barry, why don't you walk us through some of your thinking and work around the steroid implants and long-term steroid use?
Yeah, so that was another big part. When I came to UC Irvine, honestly, I was happy and working and collaborating and doing things, and I had a lot, but I was feeling honestly, a little frustrated. I wanted to do more, and I was thinking of leaving. I was actually in big conversations with David Boyer at the time, and Gary Thomas, may rest in peace, about leaving UCI and joining them up in LA. And Chris Kenny and Tony Nesburn moved their labs to UC Irvine. And I always love to acknowledge Chris Kenny, may she rest in peace. She sadly passed away about a year and a half ago. But she came to UC Irvine, and we started sharing the lab and ideas, and we did cell culture work, and we developed together this portfolio of work that was super inspiring about steroids before even my work with Ozurdex and it was why I got involved with Ozurdex, because I became super interested in steroids through my collaborative work with Chris Kenny. And we studied what steroids do on retinal cells and culture, upregulating, downregulating, tight junctional proteins, all that stuff, and published extensively. All that work actually became my AOS thesis and got all published in the Transactions of TAOS. So I was very enthused and interested in how steroids control permeability in the retina, both clinically as well as in my laboratory, and got very involved with the ocular drug delivery system from a company called Oculex. So I've acknowledged Chris Kenny, and she is still somebody I think of all the time and mourn her passing. But now I want to acknowledge somebody else that we collaborated with, and a dear friend of yours as well, Mark Blumenkrantz, because Oculex was up in the Bay Area, and Mark and I became the two key collaborators with Oculex that developed the Ozurdex, the PostureDex implant that was known, the dexamethasone implant. Mark was on the board. I was kind of more working with the vice. He was, as usual, Mark was working with the CEO and on the board, and I was working with the vice president of the company and developing the clinical trials. But that seminal clinical trial was developed by Mark and me in collaboration, and we eventually brought on Julia and Haller and George Williams, great friends as well. And so that was where, ironically, I had nothing to do with. Allergan was still 10 minutes away, and I'd done nothing with Allergan because they really had nothing in the retina space. But I helped develop thoughts about that DEX implant and the clinical trials. I sort of ran those clinical trials for them. They were a small company and helped with the running. There were people there, but I was on the phone daily with them. And then eventually when it got bought by, I forget the intermediary finance company, some private equity group bought them, and then they sold it to Allergan. And that's when suddenly I started collaborating with Allergan on a daily basis, as well as we continued to develop the Posture Dex, as it then got renamed Ozurdex at the 11th hour because of a name confusion over another drug. So that was a very exciting time, and I loved the collaboration with working with Mark on that. Mark was an amazing person, again, still very much here, but in retirement mode. But that was very exciting times. We spent a lot of time together working on the development of the Ozurdex implant.
Well, working with Mark is a treat for all of us,
Yeah.
and always has been, and so is working with Allergan. And I credit you with turning the Titanic, changing the culture at a big, giant behemoth like Allergan and helping them focus on the back of the eye really made a lot of difference for a lot of people in this world. And maybe you can explain to our listeners how amazing it was to go to Allergan and work. They had basic science labs, they had preclinical animal models, they had a huge fleet of fantastic scientists, from geneticists, data scientists, pharmacologists, and I'm sure names and faces are popping. And then they had great leadership, which ultimately was Scott Whitcup, who's
Yep.
another great seminal force in moving our field forward. But just talk a little bit to our listeners about how one navigates interacting with a for-profit pharma company or big biotech company, or a company transitioning from biotech to pharma, and keeps them, they're a very ethical group, and most of them are, but you played a role in making sure the questions asked were not just from a pure mercenary drug development standpoint, but really scientific. Speak to that a little bit, if you might.
Sure, it was a very positive experience. Again, it's interesting because I consider myself very independent, very outspoken. But I've had these interactions with drug companies my entire career that I have to say have been overwhelmingly positive. There's been a couple bumpy moments where they're thinking that they want me to say something. I say, sorry, I can't say that because I don't believe in that. But by and large, it's been a thoughtful, coherent process. And certainly it was back in the day with the companies that were developing the products, the Gansai for implant, Gilead Sciences, which is still around, and they had Cidofovir. But then casting forward to Allergan, that was a very positive experience. It was run by Gavin Herbert back in the day, and he was the godfather in a certain sense, of Orange County and Irvine in particular being, we are certainly one of the 7 or 8 biggest biotech corridors on earth. But what we're number one in is ophthalmic tech. And that was all because of 2 people, really, Gavin Herbert and his vision as a leader. Whenever there was executives at his company that had ideas that he didn't think was in alignment with Allergan, he encouraged them to go out and found their own companies. And so there's a network of people that are all former executives at Allergan that are CEOs of their own companies here in Irvine. There's another one I like to give credit to, Bill Link. He was the financier because you need money to start these things. So Versant Ventures and the other things he did, he was the one that he invested in the Gansai for implant. Chiron Vision was his, and he's so again, he was another important person in creating this amazing ecosystem in Orange County of ophthalmic tech. But back to Allergan, it was an amazing experience. So just to give people a context, here I was a full-time professor at UC Irvine, busy clinical practice. I had a lab here, but every Friday I would go to Allergan and work with James Burke in particular. I want to nod him more than anybody else because he was the one I collaborated with the most. An amazing guy from British Guiana, of all places, I think it was, and a brilliant scientist. And I would spend time in his lab. I'd bring one of my fellows with me, and we'd work on projects together, either every Friday or every other Friday. We'd have lab meetings. It was a wonderful collaboration. And you're right, they had amazing resources. They had, incredible animal facilities. They had a monkey facility there, for example. So we did a ton of projects together. It was very thoughtful. It was collaborative. We picked each other's brains. it was a lot of fun. And the whole support system, Larry Wheeler was the vice president. I don't know what the term was. I still interact with him, but he was in charge of research there, and he was great to work with. There was other scientists there that were also I interacted with a lot, but that was on the scientific side. And then separately, as the Gansite for implant, as the Ozurdex implant, the dexamethasone implant became more of a reality, I also helped with the commercial development of that, weirdly in the marketing side, but ironically more with Europe, as it turned out, than the United States. They had a brilliant and a very atypical executive named Gunilla Norgren that was based originally from Sweden. She had worked at Pharmacia. She had a PhD in pharmacology. She was the one that led the development charge in Europe, and I was somehow tasked with being the one, because of my research in steroids, again, these serendipitous things that link up, I would become what knowledge as a steroid expert and how it works in the eye. And so I was the one, before there was ready to launch the Dex implant, the Ozurdex, I was the one that helped her put together this team of KOLs in Europe, that I would be the one that would present some research, some science, to give them a context, to be excited about the Dex implant when it became available, about the role of steroids for things like diabetic macular edema, which was the focus, of course. So there was a lot of pieces to that interaction with Allergan in particular, but it was all enjoyable. It was a lot of fun. It was all very ethical. It felt very collaborative and meaningful. So I have nothing but love for all that time I spent with them.
Yeah, those were really impactful interactions, and you really paved the way for a lot of people in retina to cross over from academia or private practice into interacting with these companies and moving these companies forward. And I would also echo what you said about James. What a great guy, and infectious smile, and just never ceased to amaze me. He'd sit there and he'd look confused, and then all of a sudden he'd spew something so brilliant. And I'm sure you remember those insightful moments where you go, wow, that really makes a lot of sense. Let's look at that. Well, so those are amazing contributions. When you think back to leaving USC Doheny and taking the job at Irvine, or being at Irvine earlier in your career, and contemplating going into private practice, what were the factors that drove you to academia? And then what were the factors that kept you in academia?
Well, I always had thought I would start in academia. I mean, to a certain extent, there's a lot of factors that influence it. My father was an academician that I adored and admired. He was a professor of chemical physics at Caltech. And I always had a sort of an intellectual curiosity. Like I said, I got a PhD just for fun because I thought I had a curiosity as to how synapses were modified and how we learn. And so again, I tended to explore those intellectual passions, for better, for worse, without necessarily an end game in place. And then I went to med school without a clear definition of what I was going to do. Again, a little bit of a meandering path, also took time off to travel in between each of those. As you know, I've shared some of those stories. I would give myself sabbatical years along the way because life was long and I was not in a hurry. And I went to Caltech, then took a year off to travel, then went to med school in Miami. Somewhat serendipitously, didn't realize that they had Bascom Palmer. I just knew that they had a program that if you had a PhD, you could get an MD in 2 years at Bascom Palmer back then. Not anymore. That program shut down. Then took another year off to travel when I was after we met each other at USC. So I've enjoyed my path along the way. But when I came to UC Irvine, again, it was to follow my intellectual curiosity. And also, I will say, again, by that point, as I shared with you earlier, my epiphany was I really was committed to taking care of the ocular complications of AIDS, but I wanted to do it in a thoughtful way. And I should also, I've acknowledged him earlier, but after I left USC, I went to Hopkins, it was supposed to be Hopkins, but then Bert Glazer and Ron Michaels left, so I followed them to Towson Medical Center for fellowship. But I'd arranged to do that was a 1-year fellowship. It was one of the last of the 1-year retina fellowships, but I arranged to do a 2nd year of fellowship with Bill Freeman in San Diego, who I met when I was at USC Doheny, because he was doing at UC San Diego, just down the road, exactly what I wanted to do. He was studying the ocular complications of AIDS in a meaningful way. And so he was kind enough to allow me to come for just 1 year. His fellowship was a 2-year fellowship. So I basically went to him for my 2nd year of fellowship for 1 year after having spent the 1st year in Baltimore. And so I learned so much from Bill as well, and I want to acknowledge him. So I really was debating when I was time to make my choice. I loved, um, the notion of David Boyer and Gary Thomas and what they were offering me. And I still was more wed to going to UC Irvine, or at least being a more traditional academic. Actually, the deal that I sort of had structured in my mind and with Gary and David was they were going to set me up in Pasadena. They didn't yet have an office in Pasadena in private practice. And I was going to apply and try to get an adjunct appointment at Caltech. That was my vision. Maybe it would have never been reality, but I thought that was what I was going to try to do. That was plan 1A versus 1B, and the other one was UC Irvine. So I ended up choosing UC Irvine. I kind of liked the smallness of the department and the university. I felt like a place where anything was possible, and that's what it's turned out to be. I never really got any help early in my career while being at UC Irvine, but I never asked for help. What I loved was that I didn't have barriers. Nobody got in my way. I was just allowed to charge forward, do what I thought was best. I didn't have any barriers, and I loved being here, and I loved, it was a smallish department. Weirdly, after 3 years of being here, my department chair, Dick Keats, who's long since passed away, went on sabbatical, and he asked me, the most junior person, to be the acting chair of the department, which I thought was kind of hysterical. But I said, all right, I'll try doing this. So I had so many opportunities. He then came back a year later and then weirdly retired. I didn't know that you could retire after taking a sabbatical. I thought you'd have to come back and work. So then I stayed on, no longer as acting chair. My title changed slightly to interim chair because I was no acting on behalf of somebody else. There was just nobody else. So I was the interim chair for 3 years. And then we recruited Peter McDonald from Doheny USC at the time to become our chair. And then he left after a few years to go be chair at Hopkins Wilmer, where he still is thriving as chair. So it was a place that was dynamic, full of opportunity. I didn't mind living by the beach. That was fun too. A great place to live and raise a family. And but I love the opportunities here, and I appreciate that. When I reached a dry patch after about 7 years or so, and I began to reconsider, had I done enough in academics? David and Gary were so welcoming to me, and we had long conversations. And David actually was trying to encourage me to stay in academics. So he said, Barry, you're always welcome to have a job here. And honestly, for years after that, before they hired anybody, they would reach out to me, said, Barry, we're about to hire somebody else, but if you're interested, we're always happy to hire you. It was super sweet of them. Gary sadly has passed away since, but it was a beautiful relationship. I admired them both as sort of older brothers. I was the oldest in my family there. I still view David as my older brother, and I acknowledge him as such. I love that guy. And yet when it came push to shove, I was on the cusp of thinking about leaving, and it would have been a great life. I really was at that point, much more thoughtful about talking to Caltech and them and what could I do, set up in Pasadena. It would have been lovely. But that's when Chris Kenny moved over, and suddenly I had new doors opened here where my lab I was kind of a thin lab, and she became much more of a robust. She was more full-time in the lab. So I had an amazing lab mentor. She's an MD PhD, was an MD PhD, a general ophthalmologist, but had a robust research portfolio. And that's when I fell in love all over again with academia and research and the opportunities. And that's when I told David and Gary at the time that I was going to stay. And that's when they kept the door open for me in case I reconsidered. But I've been happy ever since, now 32, almost 33 years into it. Been a happy ride.
It's an amazing career arc. And when we think about career arcs, you've shouted out a lot of your mentors. One of the really big ones for both of us, you haven't mentioned yet, it was Steve Ryan. And tell us a little bit about your recollections of interacting with Steve. Sure. I remember multiple morning rounds with Steve when he would come over to the county and both inspire and put the fear of God into us at the same time. He was very JFK-esque with his charisma. But talk about your recollections of working with Steve and how he shaped it. And paradoxically, over your right shoulder, I can see the Ryan textbooks.
Yes. So again, completely agree. And I would almost reference him, but I felt like I'd done too many asides. But I was just honored, and I'm grateful for the honor as the Irvine Memorial Lecturer at Doheny UCLA. It used to be Doheny, of course, USC, and it's ironic because I'm at UC Irvine and I live in Irvine, California, but that is a different Irvine. That's the Irvine family not the Irvine family that founded this city, but rather founded Doheny. Sandy Irvine and his, again, there's many Irvines involved. John Irvine is a current Irvine at Doheny UCLA. But back when Doheny was founded, again, Doheny, just to reference that, is Estelle Doheny, the family fortune of Doheny Point and Dana Point. There was used to be down here in Orange County. That's our Atlantic Richfield Corporation, now known modern as Arco, the big gas company. That's the Doheny money, and there's still the Doheny Mansion on, I think, West Adams in LA. But Sandy Irvine had founded, I think it was as more as a pathology lab at one of the hospitals downtown LA, St. John's, something like that.
Vincent's.
And what's that, St. Vincent?
St. Vincent's, yeah.
Yeah. And back in the day, amazing, they recruited Steve Ryan, who never did a retina, it was at Hopkins, it was the chief resident, never done a retina fellowship. He did a pathology fellowship at AFIP, the Armed Forces Institute of Pathology, came to Doheny and turned it into a powerhouse. What an amazing human being. And again, very JFK with his charm, his boyish smile and charm, and he could be harsh, but he was brilliant and amazing and charming, charismatic, and created Doheny into the powerhouse it is today. So nothing but love and admiration for him. And he was a mentor to us all. I mean, my two mentors, and when I gave this Irvine Memorial Lecture just a few weeks ago, I think 4 weeks ago, was that recent, March 29th, I think it was. My shout outs went out to the Irvine family, but I also made sure I'm honored Steve Ryan. I acknowledge Steve Ryan, also Ron Smith and another amazing person there, and Alfredo Sadoun, my own personal mentor, and Narsing Rao. Those are 4 people that I admired above all and had amazing relationships with during my residency time at Doheny. It was that sort of place where 4 different faculty I viewed as incredible mentors, sort of friends, meaning I'm a baby resident there on Mount Olympus, but I admired them all. Steve Ryan was a transformation. I mean, he was an advisor to presidents. He was the dean of that medical school. He was an amazing. He was remarkable, unlike almost anybody else we know in academia. So yes, he was transformational and influenced my career in dramatic ways. Actually, ironically or not ironically, I referenced going to be with Ron Michaels and Burke Glaser at Hopkins. It was all because of Steve Ryan. He made that happen.
Yeah, as he did for all of us.
Career.
As he did for all of us. A quick note from Steve opened a lot of doors for all of us, and the litany of people in our field and our cohort in particular really were catapulted forward. And I think Steve was greatly influenced and helped and collaborated with Brad Stratzma, who was across town and founded Jules Stein Eye Institute. And Brad had a very similar arc, Navy SEAL, super charismatic, AFIP, pathology, came to the west side of Los Angeles, for those of you who aren't familiar with our geography, whereas USC Doheny was on the east side of Los Angeles and quite far apart, both as the crow flies and time-wise now more than ever because of our wonderful congestion. But really, really privileged to have been around those people. Now, one other theme that's sort of percolating through your comments, Barry, is the theme of integrity. And, you know, in our field, everybody's smart. Table stakes are smart. You don't get to be a retina surgeon or a wannabe retinal surgeon if you're not smart. But what has really catapulted you, in my view, is your never-ending, super strong integrity. And all of the mentors you've mentioned and all of the interactions you've mentioned lay on this foundation of integrity. And our friend Mark Blumenkrantz always uses the aphorism, integrity is destiny. And so as you've built this, what is now a powerhouse department at UC Irvine, which you really built it. I mean, there have been some great previous chairs, including Peter, but you've really taken it to the next level over the last decade or whatever it's been, where you've really been in the driver's seat completely. Tell us about how integrity has played into your decision-making.
Well, thank you for your kind comments. I'll accept them in the sense that I feel the same way, meaning to me, it's about integrity. All those interactions with pharmaceutical companies, as I already shared earlier, was always done in a way that I felt that I was still adhering to my principles. I was always vocal. I've sat on many advisory boards with you, and I'm never shy about giving my comments, and I've never pretended to be a yes person. If they wanted a yes person, they needed to go to somebody else. And I never felt that it was useful for them to have a yes person. They needed to hear my version of reality. It may not be what they perceive or view, but it was important to hear divergent or honest and candid points of view. So I've always been a bit amazed, quite honestly, that is because I'm relatively outspoken and I try to be true to my principles, and I don't feel like I've ever sacrificed them. But I've survived so long in a sort of in a bureaucratic administrative state, i.e., the University of California, the biggest and most famous public university on earth. We all have different silos. You're at UCLA, or was it for a long time? I'm at UC Irvine, but it's a place where there is a bureaucracy, and yet I've been outspoken, as true as I can be to my principles, and have still thrived and grown here. So I'm grateful to the system, but I believe in what you say. And as chair, that's been my totem. I mean, the part of why we've grown so much, I view it as my absolute guiding principle, is to be a good steward to everybody in my department, every faculty member, every trainee, every doctor, every MD, every PhD, every trainee, every faculty. My job is to treat them fairly and candidly, try to elevate them, facilitate their path, elevate their careers. And if I'm not doing that, I'm not doing my job as chair. And I don't view it as a sacrifice. I don't view it as anything but my reason to do this job. Otherwise, what's the point of doing it? The growth that happens secondarily is secondary. I'm not trying to grow this department or elevate this department. I'm trying to treat everybody fairly and kindly and support them so that they can do their best work. And so that trust to do that is based all on integrity. And I think people respond to that. They see it, and they, again, I'm not here to claim any credit for myself. That's the last thing I need. I don't need a smidge of an honor or a credit, but I want to treat people fairly always. That's my guidepost.
And that's the reason why you're attracting such a great team and continue to recruit and attract great team members. When you look at a potential resident, or more germane to this conversation, fellow or junior faculty member, what are you looking for? What's the secret sauce, the cultural fit? What are the characteristics, besides how smart they are and how productive they are and whether their work is a fit for your department, which is sort of true of every job? What are you looking for when you're talking or looking at a person?
Well, each of those are a bit of a different set of criteria in a certain sense. And of course, where I can really focus on what I'm trying to do is with the faculty, because the residents and fellows end up coming through a match. So all you can do is list your priorities, your rank order, and then the system decides where you're going to match. Whereas when I'm recruiting faculty, it's very bespoke. It's very one-on-one, with very close to what I'm looking for. But you've highlighted most of the features. I want them to be smart, but to get to this point, they're smart. I want them to be collegial, collaborative, have integrity, be kind, caring. That's particularly at the resident and fellow level. The faculty, I'm assuming a lot of that, but at the faculty, I start looking at more what are their intellectual interests. I began to branch out more, but the presumption is still what I'm looking for. I look at every resident and fellow and faculty as a family member. And so how are we going to live together? How are we going to work together? We don't have to play together, though we tend to interact socially as well. Never forced. It's always the option of people. But the faculty in particular gets together on a regular basis. We have our social director, Mithil Mehta, and others, so that he's always making sure that we're getting together next Thursday night at a restaurant to sit around and have dinner and maybe some drinks along the way. So we try to be collaborative, but I'm looking for thoughtful people that have a worldview that they want to make a difference. They want to help us change the world together, take good care of patients, and think in a visionary way of how we can make this a better place. We want to leave a mark together on this world by leaving it better than what we started with. And that's what I'm looking for.
So if you were a junior fellow or senior fellow somewhere out in our global audience, whether it's, Brazil, a country that you're a native of, so to speak, and or whether it's India or Mexico or Japan or wherever our listeners are, Israel, what's your advice to how to choose a career path and then how to optimize?
Yeah, it's an interesting question. It's because, ultimately, of course, most people go into private practice. That's where the jobs are. And they do it in different ways. Some like to be a small community practice. Others want to have a more academic private practice. But for those that are choosing an academic career, again, there are several different paths for that as well. But mostly it's it's again, there are and many ways to do that, but I think the motivating factor, particularly for the academics part, but many community academic uh, private practices, is the teaching aspect. You've got, I think, a big driving factor for us is the desire to share knowledge, and we do that in many ways. But I think that's what you want to manifest is a passion for teaching, but also an intellectual curiosity. Because in our department, there's these very serious, much like there is at UCLA, but many of my faculty are what are clinical professors, and there's not a requirement for any research for promotion in that series, at least by the university standards. But in our department, we hold people to a higher standard. We expect people to have scholarly activities. So you have to also enjoy asking questions, thinking about your patients not only individually but also categorically. How can you make things better or different? What are the research opportunities? Not think of your patients as research opportunities, but think of their problems and is there a common theme? Is there something we can learn from them so that we can be even better doctors? And so having that intellectual curiosity, I think that applies both to private practice and academics, but I think that's what should be the driving force, is not only being the best doctor you can be, but maintaining your intellectual curiosity. It makes life fun. It makes your career more interesting. It makes things. I never felt like I had a tedious day because I spent all day long, every day asking questions. And that's what made every day fun. I wake up every day happy, looking forward to going to work every day, looking forward to coming home, the happy home life as well, because
that's genetic, Barry. That's genetic, and we're going to come back to that in just a second because you are over expressing the happiness gene, which makes us all envious.
Yeah, I view it as a chemical imbalance I have. I wake up every day happy, and I have no idea why.
Isn't that wonderful? So I was going to just refocus that answer for a minute. So it sounds like synthesizing your experiences and your culture there. If you were advising a person early in their career, it sounds like you're telling them to introspect and figure out what it is that drives them and what they're looking for. And once they've realized where they fit culturally in the structure of their own country's medical delivery matrix, then target those areas and to be true to themselves so that they can end up with a good fit for themselves. Does that sound about right?
I think you've taken the question differently than the way I did, but you're right. Back to focusing on the question that you asked, 100% agree with you. I was then taking it the next step, that in an academic career that you need to make sure you keep your curiosity hat on. I think that's good for everything in life. Always asking questions, finding yourself in the middle of all this, of course, finding something that's a good fit for your personality in a situation that's good for you. But when you're in that fit, making sure to never stop asking that question, why, and how you can make things different or better. That's the fun of life, is to try to make a difference.
So with that in mind, and if this is too personal, forgive me, but can you just tell us a little bit more about your family, your father, your mother, your siblings? It's such an amazing story, your dad in particular, but your sibs are amazing as well. And I just thought it would be worth hearing about not only their accomplishments, but really what you took from your dad's life of inquiry.
Well, thank you for mentioning him, and I'm always happy to honor him. I have a photograph of him right here next to me. He was an amazing person. So my family were Jewish, Ashkenazi Jews from Europe originally, but over 100 years ago, we're a branch that ended up in Latin America, in Brazil. So my parents, my grandparents' generation, as young people, moved to Brazil, again, around the time of the Russian Revolution, from Poland, Germany, Russia, etc., Ukraine. It's complicated geopolitics there. But
I now call it the Austro-Hungarian Empire because it's a big map, a moment where there's a big circle.
So Ellis Island was open at the time, but everybody went, imagine the time of change. People were leaving their circumstances and going off on their own with 2 nickels maybe to rub together in their shoe at age 18, off to the new world. Amazing leap of faith. What a time. And Ellis Island was open, but everybody went where they knew somebody. And my family, the various branches, knew somebody in Brazil. So both sides of my family, they immigrated from Eastern Europe and Central Europe to Brazil in the early 1900s. And all my extended family is still there. My parents grew up in Brazil, born and raised in Brazil. And my father was an amazing person. He grew up in Brazil and was a brilliant college student. Those days you had to take an entrance exam to get into college. And he was number one at the University of Sao Paulo, biggest university in Latin America or Brazil. And then he graduated 4 years later in engineering, number one in his class. But he was an intellectual curiosity, and he wanted to come to America, or wanted to explore his intellect. And he was interested in radiation chemistry at first, and he went off to Scotland. He was in Edinburgh, did a fellowship, and then he wanted to go to grad school in the United States. And this is the way I tell the story. I'm not sure it's exactly right, but even though he's a nice Jewish boy, Brazil is the world's largest Catholic country. So back in that era, they used to send their best and brightest not to Harvard, but to America's greatest Catholic university, Notre Dame. So my father came to Notre Dame to get a PhD, which in physical chemistry, and my mother came with him. They were married at the time, young couple. She was not allowed at Notre Dame at the time. They met in the Department of Chemistry at the University of Sao Paulo. She had to go to St. Mary's because Notre Dame at the time was still not co-ed. That happened later. And then my father got his PhD in all of 2 years at Notre Dame. This Brazilian kid out of nowhere went back, and I was born just a few months before he got his PhD. So I was born in the United States, but we moved within a few months to Brazil, where we're going to live the rest of our lives. My dad was going to be a professor at University of Sao Paulo, Department of Chemistry, but he got frustrated after a couple of years. So when I was three and a half, we turned around and moved back to the United States, to the University of Illinois, Champaign-Urbana, where he was recruited to, again, Department of Chemistry, physical chemist, where my sibs, Miriam and Nate, were born. And then eventually he was recruited to Caltech, where he spent the rest of his career, where my youngest sister, Sharon, was born in Pasadena. And he was an inspirational figure. My parents were both lovely people. My mom ended up being a high school chemistry teacher at Westridge School for Girls, a private high school in Pasadena. She was the head of the science department. My dad had a thriving career initially as a physical chemist at Caltech, and he morphed into a chemical physicist because he started doing complex quantum mechanics and learned Mathematica by Stephen Wolfram, this complex coding language that explains fractals, when he was in his 80s, for God's sake. He worked at Caltech until he was well into his 80s. And he had an amazing career, and he never asked his children to go into science or to pursue academic careers. He was just led by example, and we admired him. And so I became first a PhD, and that was a lovely time. He was at Caltech, I was at Caltech in the next building over. We used to have lunch every day at the Athenaeum, the faculty club there. My sister, who came along next, got a PhD in molecular biology at USC and is a researcher, the head of research for many years at the Department of OBGYN at UCSF, where, as a small coincidence story, the person that hired her, I forget his first name, but I know his last name, was a Dr. Jaffe, the father of our very own Glenn Jaffe, hired my sister to her job that she still holds all these years later in the Department of OBGYN at UCSF. And then my younger brother, Nate, was an MD, went to Stanford, then UCSF, and he was the chairman of the Department of Emergency Medicine for 18 years or so, led that unit, and he recently left that. Again, pretty advanced in his career. He's now the chief academic officer for Washington National Children's Hospital in DC, and the chair of the Department of Pediatrics now. He was chair of emergency medicine. He was dual trained, and now he's chair of pediatrics at George Washington. So kind of a remarkable academic career for all of us. And our youngest sister, Sharon, has Down syndrome and has been a passion for all of us and a love that we have for her. And I still visit her and my mom every Saturday. They both live in Pasadena. I still drive up from Newport Beach every Saturday to have lunch with the both of them. So
Your
that's our family.
your family is an inspiration, and I would just say this about Nate's career move and correct you on one thing. It's not late in his career. If we use your dad by any metric, it's about
Right.
halftime for Nate.
Fair
So
enough.
yeah, I think he knew what he was doing. Well, Barry, thank you for sharing that with us. And I want to close on something speculative, and that is as you sit at this pinnacle of innovation at the crossroads of biotech and retina there in Orange County and ophthalmic technology, you see so much going on. And without mentioning any companies or startups by name, that's not what I'm looking for. What I'm looking for is predict where you think the next big innovations are going to be in retina. Is it AI? Is it cell therapy? Is it gene therapy? Is it big data? Where for you? What's exciting you? I mean, they're all going to make giant
Right.
contributions. They're all going to take some time. But what is it for you that's really got you excited as you look down the road in our field?
Well, it's a great question. I think everything you've already pointed to are going to be huge contributors, as you mentioned. But I'm particularly excited about the changes that we see coming in cell therapy and gene therapy. We actually have part of this glorious, fun time I've had at UC Irvine. Again, they keep giving us gifts. There's a lab building right outside my window that they're just about done with. We're going to move into it. They've given us the sixth floor, which is the penthouse, 30,000 square feet of lab space. We have amazing researchers, but we're developing a program in genome editing here in the Department of Ophthalmology, but it's really a gift to the School of Medicine because we're not even hiring eye researchers. We just hired Audrey Lapanaiti, trained with Jennifer Dowden at Berkeley, the Nobel laureate, and she works on various diseases, but has nothing to do with ophthalmology. We've asked her now to start focusing on ophthalmology and genome editing. I think that's a huge transformation of what's going to happen, but also the various cell therapy approaches I think are going to be transformational. I think we're going to get from the era, I love the era of pharmacology that we're in drug delivery, and I'm excited about that, but we're going to then go into ultimately cell therapy, and hopefully we'll get to something closer to regenerative medicine that will really be transformational, along with the gene therapy and the editing that we can do to really get in the era of one and done therapies where we can really get to the point of transforming, restoring people's vision after a lot. And that's going to be a mixture of genome editing and cell therapy. I think that's ultimately the long term. There's a lot of challenges and hurdles. Actually, I just realized Audrey Lapanaiti has been working in cystic fibrosis. I was trying to remember what she did. And we recruited her to our Department of Ophthalmology because genome editing is agnostic to the disease. It's CRISPR-Cas9 technology that she's bringing to us. So again, there's so much exciting stuff going on that I think that's the direction we'll be going primarily.
So if you're listening to this podcast, you can hear or see the excitement in Professor Barry Kuppermann for the future of our field. And Barry, I want to thank you for sharing your story. You've been extremely successful and fortunate in that you've been able to make contributions that have combined basic translational and clinical impact. And you felt that firsthand. And I think the reason that I've always been inspired by your work is how dedicated you are to relieving the suffering of each individual patient that you take care of, and treating your faculty as family has built this remarkable institute at Irvine. I'm going to overstep my bounds here and invite our listeners to reach out to Dr. Kuppermann and visit the Gavin Herbert Eye Center in Irvine, California. If you're traveling to California, it's one of the great places to visit. And if you're coming to the ASRS meeting in Long Beach, I recommend strongly that you introduce yourself to Barry. He's one of the nicest, most affable people in our field, and always interested in meeting young people from foreign lands. And by foreign lands, I mean anything that's not inside the Southern California circle here. So, Barry, thank you. Thank you very much for sharing your journey with us today on Retina Rounds. Barry, that was fantastic.
If you enjoyed this video, please visit us at RetinaRounds.com. There you can sign up for our email list. You'll get a notification every time a new video is posted. And if you have an interesting video or a tip or trick that you'd like to share, please follow the links on our website and you can upload your video there. Thanks so much for watching.